Hello guys, welcome to the first videos of bacteriology. So what is bacteriology? Bacteriology is the branch and specialty of biology that studies the morphology, ecology, genetic, and biochemistry of bacteria as well as many other aspects related to them. So, bacteria are also called as microbes. So, if you're wondering what are called microbes, I've got a video on introduction to microbiology. So, I have explained in that video what are microbes. So, you can also check that one. Bacteria are prokaryotic cells because they are unicellular. They play important role in disease and health. So, you might be thinking how they are going to play a role in health because they are part of human microbiota, or we can also call it microbiome or normal flora. So, bacteria have less complex structure as compared to eukaryotes. Bacteria like certain membrane-bound organelles such as nucleus, mitochondria, endoplasmic reticulum, VER, and Golgi complex. Classification. Bacteria are classified by shape into three basic groups. Number one, cocci, but in some places it is also pronounced as cocci. Number two, bacilli. Number three, spirochetes, but is also pronounced spirochete. Number four, pleomorphic. The cookie are round, as you can see in the middle. The bacilli are rods, as you can see on the left side. And these parakeets are spiral shaped, as you can see on the right side. So some bacteria are variables in shape and are said to be pleomorphic, heterogeneous shape. So for example, consider a bacteria that has got the spiral shape, the cocci shape, and the bacilli shape. So like it has round, rods, and spiral shape. So we'll tell them that bacteria are as pleomorphic shaped bacteria. So in addition to the characteristic shape of bacteria, the arrangement is also very important. Arrangement of bacteria is determined by the orientation and the degree of attachment of bacteria at the time of cell division. So let's start with the cell wall. Cell wall is the outermost component common to all bacteria except Mycoplasma species, which are bounded by a cell membrane, not a cell wall. The shape of bacteria is determined by its rigid cell wall. Some bacteria have surface features external to the cell wall, such as capsule, flagella, and phyla. And then location. Cell wall is located external to the cytoplasmic membrane and is composed of peptidoglycan. Peptidoglycan provide structural support and maintain the characteristic shape of the cell. So, the cell wall has several other important properties like in gram-negative bacteria it contain endotoxin, a lipopolysaccharide. Cell wall polysaccharide and protein are antigen that are useful in lab identification. Prion protein play a role in facilitating the passage of small hydrophilic molecules into the cell. Prion protein is the outer membrane of gram-negative bacteria, act as a channel to allow the entry of essential substances such as sugar, amino acid, vitamins, and metal, and as well as many antimicrobial drugs such as penicillin. Gram-negative bacteria have a complex outer layer consisting of lipoprotein protein in the outer membrane of gram-negative bacteria, act as a channel to allow the entry of essential substances such as sugar, amino acid, vitamin, and bacteria, as well as many antimicrobial drugs such as penicillin. Gram-negative bacteria have a complex outer layer consisting of lipopolysaccharide, lipoprotein, and phospholipid. So now, Let's look at the cell wall of acid pass bacteria. They are unable to be gram-stained. They are called acid pass bacteria because they resist decoloration with acid alcohol after carbon for Paschen staining. And this is due to the fact that they have the properly related to high concentration of lipid called mycolic acid. And the acid fast bacteria also stain red with methylene blue. So bacteria, the Mycobacterium tuberculosis is acid fast bacteria. Nocardia asteroides is weakly acid fast bacteria. So the meaning of the term weakly is that if the acid polystyrenian Mycobacteria, the Mycobacterium tuberculosis is acid fast bacteria. Nocardia asteroides is weakly acid fast bacteria. The meaning of the term weakly is that if the acid polystyrene process uses the weaker solution of hydrochloric acid to decoloration, that used in the stain for Mycobacteria, then Nocardia asteroid will not decolorize. However, if the regular strength hydrochloric acid is used, Nocardia asteroid will decolorize. So we are there talk about the structure outside the cell wall, which include capsule, flagella, phyla, or fibri, and the glycocalytes. Let's start with the capsule. So capsule is a gelatinous layer covering the entire bacterium. It is typically composed of polysaccharide. The sugar component of polysaccharide vary from one species of bacteria to another. Capsule is important for four reasons. Number one, it is determinant of virulence. So what is virulence? Virulence is the severity. Number two, species identification of an organism can be made by using anti-serum against that capsule polysaccharide. Number three is the capsule polysaccharide are used as antigen in certain vaccine because they are capable of eliciting protective antibodies. Number four, the capsule may play a role in the adherence of bacteria to human tissues, which is an important initial step in causing the infection. Next up is flagella. As you can see, this long hair of this green bacterium, are the flagella. Flagella along with appendages that move the bacteria toward nutrient and other attractants. A process called chenotaxis, the long filament which acts as propeller, is composed of many subunits of protein. Flagellum arranged in several intertwined chains. Phyla or fibri. Phyla are hair-like filaments that extend from the cell surface. They are shorter and straighter than flagella. Composed of subunits are phylum, a protein arranged in helical chain. They are found mainly on gram-negative organisms, which have two important roles. They immediately the attachment of bacteria to specific receptor on the human cell surface, which is necessary step in the initiation of infection for some organism. So for another role is a specialized kind of filler that is the sexual filler that form the attachment between the male that is the donor and the female that is the recipient bacteria during conjugation. So next up is the glycocalyx. It is also called the slime layer. Glycocalyx is a polysaccharide coating that is secreted by many bacteria. So it covers surfaces like a film and allows the bacteria to adhere firmly to virus structure of, for example, skin, hot wall, prosthetic joint. So glycocalyx is an important component of biofilms. So bacteria spore. This is the last structure that we're going to talk about, and it is a small and bacteria spore. This is the last structure that we're going to talk about, and it is a small and oversafe structure. It is highly resistant. So these highly resistant structure are formed in response to adverse conditions. Adverse condition can be like the changing in pH, changing in temperature, or maybe certain other environmental conditions that are not good for bacteria. So it can convert into its poor form. You know what, guys? Sulfose contain bacterial DNA, ribosome, and other essential components like cytoplasm, peptidoglycan, very little water, and most important, the keratin-like code that is responsible for remarkable resistance of the spore to heat, dehydration, radiation, and chemicals. So, spore has no metabolic activity, which meant that it can remain dormant for years and favorable condition like good amount of nutrient, water, and enzyme will break the code of these four nutrients, and water will enter the spore will differentiate into the bacterial cells.